Hello, everyone, and welcome to the NOAA Omics Webinar Series. My name is Tracy Gill, and along with Catherine Egan, we are the co-hosts for this series. Before we get started, here are a few logistics, and most of this information is in the chat box. All attendees are muted, but you can enter your comments and questions into the chat box at any time. The presenter today is fine with clarifying questions during the talk, but prefers to take most questions at the end. We also have a few poll questions at the end, and we hope you will participate. If you want to go to a full screen or larger slide view, look above the upper right corner of the slides and find the button that has a box with a bracket around it. That box toggles the full screen view on and off. There are two ways to access live closed captioning, and they are described in the chat box. If you are interested in getting a PDF copy of the slides, a link to the recording, and possibly a summary of the chat, we will send it out within a few days after the talk to all who are registered. If you are not on the weekly NOAA Science Seminar email list, but you'd like to be, please contact me at tracy.gill@noaa.gov, and I will add you to the list. And many thanks to our captioner today for making this recording more accessible. And now I will pass the mic to Catherine Egan, the Omics Coordinator in the NOAA OAR Ocean Portfolio with the OAR Office of Ocean Exploration. Catherine is also the subcommittee co-lead for the NOAA Omics Working Group. Catherine will introduce the NOAA Omics series, today's topic, our presenter, and moderate the questions at the end. Catherine?
All right, thank you so much, Tracy, and hello, everybody. Thank you all so much for joining today. Welcome to the NOAA Omics Seminar Series. As Tracy stated, I am your co-host, Catherine Egan. I am the OAR Omics Coordinator, and I also sit in NOAA Ocean Exploration. I also sit on the NOAA Omics Working Group as a subcommittee co-lead. The Omics Seminar Series is hosted by two NOAA groups, NOAA Ocean Exploration and the National Ocean Service, through the One NOAA Science Seminar Series. Tracy Gill and I will be co-hosting the seminars today, and I will be moderating the questions towards the end of the presentation. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last several years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway, both within and outside of NOAA. The seminars from this series are made available on YouTube and posted up on our Omics website a few weeks after the initial presentation. I will drop a link to that in the chat now so you can view past seminars. Registration for our next seminar will also be posted up on this website in the next few weeks. All right, so with that, our speaker today is Dr. Subba Chaganti, and his presentation is titled Environmental DNA and RNA for Advanced Management and Accurate Surveillance of Aquatic Invasive Species. So Rao is an assistant research scientist at the Cooperative Institute for Great Lakes Research at the University of Michigan. He was also a recipient of the Early Career Environmental Research Award from the American Society of Microbiology in 2019. He is currently serving as editorial board member for the Water and Hydrobiology journals. His research focuses on understanding the structural and functional diversity of the microbial community, including bacteria, archaea, and protists, and how they mutually interact with the aquatic, terrestrial, and host-associated environments, how they are influenced biotic and abiotic mechanisms, and how they are altered by aquatic invasive species and other anthropogenic inputs. Rao's hobbies include biking and playing chess. So Rao, thank you so much for presenting today. I will now pass it off to you.
Thank you, Catherine, for the nice introduction. Thank you, Tracy, for all the logistics. I really appreciate it. Good afternoon, everybody. I'm Subba Chaganti, and most popularly known as Rao. So I'm an assistant research scientist at University of Michigan. Today I'll be presenting about environmental DNA and RNA for advanced management and accurate surveillance of aquatic invasive species. And I would like to acknowledge the co-authors involved in this study, NOAA Global Scientist Henry Vanderploeg and Nathan Marshaw as a postdoctoral fellow. and also a lot of technicians from the Cooperative Institute who helped to get the samples from the field like the mussels and stuff. With that, I'd like to start the presentation here. So, as you know, invasions are happening in the Great Lakes, elsewhere, everywhere in the world. Especially if we look at the graph here, we have on the x-axis 1998, like the years, and on the y-axis left hand, we have months, and y-axis right hand, we have the chlorophyll concentration. So over the time, the chlorophyll concentration, especially in the early months like March to June, has drastically decreased. There are many reasons, but one of the main reasons they think is because of this Dreissenids invasion. This mussel invasion has reduced this chlorophyll concentrations as they're clearing up. But it has two main problems. One, in the offshore, it's making no food available for the lower trophic level. And near shore, they're actually enhancing the harmful algal blooms that's messing up both at the near shore and offshore food webs. So changes in the phytoplankton and zooplankton abundances and community composition has co-occurred with these Dreissenid invasions. Currently, Great Lakes, especially like the Lake Huron, has experienced, as we have seen, this chlorophyll decrease drastically. So while there are several methods to assess distribution of benthic invasive species, omic tools may be able to provide a more competitive picture of species abundance and its viability. So when we say the omic tools, like when it comes to monitoring immediately, the environmental DNA strikes as it is more advancing in the past decade. When it comes to environmental DNA, like it's not just the Dreissenid mussels, it could be used to monitor any other species in this aquatic system. Like, as I said, we have more than 180 invasive species in the Great Lakes, or elsewhere we could use this technique to monitor different kinds of species. How does this environmental DNA works? So again, most of you know, like, regular metabolic activities or dead cells, or during the feces or urine, like all those many activities are during the spawning, the cells will be released from these organisms and they into the environment or the aquatic system. So when we capture the water, we get this DNA signal. When we bring them back to the laboratory, we could process the using multiple markers. Again, immediately the question arises is, which genetic marker should we use? Is it cytochrome C oxidase like COV1, or cytochrome B, or 16S or 18S or others? So it's a big challenge, like, some has a good database, some don't have, some are really easy to use because they have an abundance of genetic material. So again, it's a lot of challenges, like, which one to choose, which one not to choose. Let's step back and see, like, a high school grade science, like, what is happening inside a cell. This is a typical eukaryotic cell. And you can see here the nucleus and the chromatin in it, and then we have mitochondria. Again, the number of mitochondria vary. Could be like 10 mitochondria in a cell, or it could be 1,000 mitochondria in a cell. Sometimes it could change from species to species, and also the life stages. So we have like two different genomes in the cell. One is a mitochondrial and another is a nuclear. So which one is better for us for monitoring purposes? Is it mitochondrial or nuclear? Let's look into a little more details of what is the differences and similarities between them. When we see the mitochondrial DNA, it's really small, like approximately 17 kilobase pairs. And its DNA is in circular shape. As I said, it has huge range. It could be ranging from 10 to thousands of cells per cell. Sorry, 10 to thousands of mitochondria per cell. Whereas its counterpart, it's nuclear, it's only one per cell. However, it's huge in size, like 3.3 billion base pairs approximately, and its shape is mostly like a linear in shape, whereas its mitochondria is circular in shape. So now we know we have like both mitochondria and nuclear. Mitochondria is circular, maybe a little bit more stable. But overall, not just mitochondria or nuclear or selection of marker, there is one more bigger problem is it's hard to differentiate eDNA signal from live versus dead. And that was one of the major challenges in this field is how do you differentiate, because the DNA could be hanging around for some time in the environment before it's completely degraded. And sometimes it could be settled in the sediment and it could be coming up during the resuspension events, or it's actually carried over by fishing nets, or it could be like a boats, or it's a bird poop somewhere else, or like all these materials coming from somewhere else, and it could sometimes potentially lead to a false positive signal compared to the living individuals. How could we avoid, when we are getting an eDNA sample, are we getting anything additional? Can we use that for more purposes? Yes, in fact, when we collect the eDNA sample, it's not just the DNA. We have a lot more in that sample. We have RNA, we have proteins, and we have metabolites. All those things are coming along this water sample. So when we capture the DNA, if you're processing the DNA, it's generally genomics. And if you're processing RNA, it's more like a transcriptomics. Similarly, for proteins, it's proteomics, and metabolomics. That's where the name omics is like the omics is common prefix for all these technologies. And again, if you're doing like for a whole community instead of an individual organism, the meta is automatically added in the prefix. So like metagenomics, metatranscriptomics, metaproteomics, and so on, like the metabolomics. Then this could be used not just for the invasive species, but could be used for monitoring the fisheries, harmful algal blooms, like are making connections within the food webs, and this data could be used for modeling and better management purposes. So as we are capturing in the water sample also like RNA signal in addition to DNA, could we do anything better? Can we avoid this false positive signals from which are occurring from the slug DNA or carryover DNA, legacy DNA? So if you look into a little more closer of DNA versus RNA here, like DNA is double-stranded, and whereas RNA is a single-stranded, and that's why it is less stable in its state. If we go a little further within the RNA, there are three different kinds of RNAs mainly, like ribosomal RNA, which is more stable because of its structure, and then the tRNA, transfer RNA. Further, we have a messenger RNA or mRNA. So most of you know, like the mRNA vaccines now we are using for the COVID. So that's an easy thing to remember. And what are their functions, where they're functioning in, ribosomal RNA and tRNA are mostly functioning in the cytoplasm, whereas messenger RNA is functioning in nucleus and it migrates to ribosomes in the cytoplasm. What are their main functions involved? When it comes to ribosomal RNA, it's mainly involved with the structural component of the ribosomes itself, whereas tRNA helps in moving the RNA, mRNA, and then amino acids to ribosomes, whereas mRNA is carrying the DNA sequence that's actually the information to ribosomes for the transcription. So as you see, their structures, like mRNA is more like a short and linear, whereas ribosomal RNA is a complex. So that's why like the mRNA is least stable compared to other RNAs. So when we compare with the DNA and RNA, RNA is less stable, and within the RNA, mRNA is less stable. So with this information, we want to actually compare what is happening with the Dreissena environmental DNA and RNA. We want to compare how this degradation is happening across the genomes, like as we saw, the mitochondria and nuclear. And also, we want to do the size fraction. So as this nuclear material is released into the environment, they try to break down and becomes a short fragment. So we want to compare within the range, mostly use it for the quantitative real-time PCR. We generally use less than 400 base pair. Within that range, is there any impact for the size selection? And then we also want to compare how is the degradation patterns DNA versus RNA. As we know, like it's a single strand versus double strand, it's highly likely the RNA degrades faster. And within the RNA type, like is it a ribosomal versus messenger RNA, which one is degrading faster? Can we use any of these for enhancing for our predictions or for better monitoring purposes, this information? So with this, we have selected 4 different genes, 16S gene and CO1, that is the cytochrome oxidase subunit 1 gene, which are from the mitochondrial genome. Within this, the 16S is ribosomal, and cytochrome oxidase 1 is messenger RNA type. And then we have like the H2B and 18S. These both are nuclear region. H2B is a messenger RNA and 18S is a ribosomal RNA. So one thing with the H2B is it's generally is highly expressed during the S phase of mitosis, and it is predicted to be very low RNA concentrations in the dead and slugged materials, and we can actually differentiate which is a recent like live material versus a dead or slugged material. So that's the messenger RNA. And then, as I said, like, not just comparing the genomes, we want to compare the type of RNAs as well as the fragment length. For the 16S RNA, we took 2 fragment lengths. One is 141 base pairs and another one is 343 base pairs. And similarly, we took for the H2B gene, we took like the 75 base pairs and another one is a 250 base pairs. The person in the picture is Nate Marsha, who was a postdoctoral fellow, who was actually involved in the study. Currently, he moved on to Stantec as a research scientist. So here, we took several mesocosms in order to conduct the experiments. You can see one on the left-hand side, there is nothing, no mussels are there. That's a control just to see if there is any cross contamination happening. And then we have like, in addition to control, we have 5 mesocosms, although here I'm showing only 3 mesocosms in addition to control. We increased the number of individuals. Here n is equal to 2, which means we have 1 zebra mussel and 1 quagga mussel. Similarly, we have n is equal to 6, 12, 24, and 48. Those are the equally distributed zebra and quagga mussels. And then we incubated them in the mussel medium for 24 hours. So after 24 hours, we assumed some nucleic acid materials like both DNA and RNA released into the water. At that point, we removed the both zebra and quagga mussels from these mesocosms. Again, these mesocosms was run in triplicate, so we have in total 18 mesocosms running parallelly at the time. So after removing these mussels from the mesocosms, then we monitor for nucleic acid degradation. So here you could see like the mesocosms was removed. So that's the time 0 we considered. So after the 24 hours mussels were there, we took off the mussels. That's where we considered the time as a 0 time for degradation purposes. And then we monitored over like 240 hours, which is like 10 days. So after completion of the monitoring, samples was collected. The samples was immediately preserved in the minus 80 degrees centigrade. The samples was filtered using glass fiber filters, and the filters were stored at minus 80 degrees centigrade. After the experiment is completed, we extracted the DNA and RNA using a commercial kit, like simultaneous extractions was done for both DNA and RNA. And then RNA is converted into complementary DNA, a single strand, that is cDNA. And after those conversions, we used 4 different markers, as I mentioned before, like 16S, 18S, CoA1, and H2B, to analyze or to quantify them using quantitative real-time PCR machine. So after the results were obtained, we converted them into log copies per ml and plotted over the time, like x-axis you could see here. After plotting, we also converted, or we made a ratios log ratio from RNA to DNA, and then we compared. Here you could see this is our prediction is in the time 0, RNA might be in higher concentrations. As the time declines, RNA might be degrading faster compared to DNA. And based on that ratio, we could predict if DNA is freshly released from the organism, or it's like few hours old or few days old. Based on that, we could be able to predict. So here is the initial concentrations. When I say the initial, that is the after 24 hours when the mussels were taken out from the mesocosms, that's the time 0. We monitored nucleic acid concentration, all the abundances. On the x-axis, you could see the number of mussels, like 2 to 48. And on the y-axis, we have initial concentration, that is after 24 hours log copies. This is 0 to like 10 to the power 12 log here. So you could see here from 2 to 12, the concentration has increased, but they range between log 6 and log 8 cells. And in this plot, in the previous one, we showed like the total nucleic acids. Here we are showing the nucleic acid concentration for each gene that we monitored, both for DNA and RNA. On the x-axis, you see the nucleic acid concentration, log copies per ml, and on the y-axis, you see the genes, and with after the underscore, the number indicates its fragment length. For example, here we have 16S, that is the gene, and 141 is the base pairs. Similarly, like the 341 here is the number of base pairs of the fragment size. And then when it comes to the plot, you could see here the circle indicates mitochondrial material and the triangle indicates nuclear material. If it is a closed circle or closed triangle, it indicates DNA. If it is an open circle or open triangle, it indicates RNA. And if you see a symbol beside the triangular circle with R or M. R indicates ribosomal RNA, M indicates messenger RNA. And then if you see this point here, this is the mean, and the bar, like the color bar, is nothing but the standard deviation here. Here you could see all the highlighted portion, like the 16S, short fragment, long fragment, and CO1. These are all the mitochondrial genome coming from, and their DNA concentration is roughly like 10 to the power of log 5 cells. When we compare to the nuclear DNA for the 18S and H2B, it's much higher than the mitochondrial DNA. When we compared within the 16S short fragment, that is 141 base pairs, from DNA to RNA, you could clearly see RNA has a really high concentration. It's more than double the concentration compared to the DNA. Similarly, for the 18S, you could see the DNA concentration is almost like less than half compared to the RNA concentration. Again, these two are the ribosomal. This is the RNA, and this is DNA concentration here. So in both cases, for mitochondrial and nuclear genomes, like the RNA concentration is higher than the DNA concentration for 16S and 18S genes. Similarly, we compared for messenger RNA versus the DNA. In this case of CO1, you could see again the RNA concentration is higher, but it's not as like ribosomal, like it's not quite high as a ribosomal one. Interestingly, when we noticed a H2B gene, RNA concentration is really low compared to its DNA concentration, very, very low. In fact, like we weren't able to notice for some of the samples. So the reason behind is, again, H2B gene is only expressed, this is messenger RNA, is expressed only with the active cells. They're mainly involved in the S phase. So active divisions, that's when the H2B gene could be available, but it's not present in the, or it's not expressed, it's present, but it's not expressed in the sluggish cells or dying cells. So after that, we actually calculated the half-lives for these different gene markers. So how do you calculate? What is a half-life? It's like the amount of the time required to degrade 50% of the material from the initial is a half-life. So over the time, as we monitored over the time for 240 hours, we calculated the half-life for each of these genes. Here again on the x-axis, we have like all the different genes, and with this fragment size, you could see the base pairs. On the y-axis, we have time, half-life in hours, like 0 to 20 hours. Here, the mitochondrial on the left-hand side, you could see there is not much significant variation. There is variation, but it's not significant variation within the mitochondrial. Whereas when we compared nuclear to mitochondrial, we have noticed a significant variation between their half-life times. So the nuclear half-life time is much shorter compared to its mitochondrial. Although if you notice in the previous slides, the nuclear DNA is actually higher in concentration compared to mitochondrial, but the degradation for mitochondrial is longer compared to nuclear. The reason could be is mitochondrial DNA is in circular shape, whereas it's a nuclear is more like a linear. So that could be one of the reasons for difference in the degradation pattern. And then we compared length. Here we have like 16S 141 base pairs, 16S 341 base pairs. So if you see, we slightly noticed a change in the half-life time between the two, but again, it is not significant. When we compared for the H2B gene for the 75 base pairs and 250 base pairs, we haven't really noticed any variation between those two. Next, we compared with the eRNA for eDNA. You could see there's a huge difference. The half-life time for the eRNA is much shorter compared to DNA. And within the eRNA, again, they followed the same pattern as eDNA. Mitochondrial eRNA has a longer half-life compared to its nuclear eRNA. Here, what we have done is we calculated the log ratios for short versus long to see if it is following a linear pattern or not. The 16S mitochondrial rDNA here, again, like the x-axis is time since the removal 0 to 240 hours, whereas the y-axis is the ratio. There was no real correlation between them, the short versus long. Same thing for the H2B. The correlation was really low. Further, we have done like a ribosomal versus nuclear material, that is the RNA to DNA ratio. Again, like the y-axis is log of ratio, RNA to DNA, and x-axis is time since the removal, that is after 24 hours the muscles was removed, considered 0, and we went all the way to 240 for all three. In the plot A, you could see 16S mitochondrial RNA, and B, we have a CO1 mitochondrial mRNA, and C, we have 18S. Again, the ratio is for both comparing the DNA versus RNA. If you see the first graph on left-hand side for the 16S, you could see at the time 0, the ratio is really high, it's close to 3, whereas as the time goes on, like the ratio is declining. That clearly indicates that the RNA is degrading much faster, and you could actually tell based on the ratio when it is released. Similarly, for the CO1, the same pattern followed as the 16S, and 18S also the same pattern. However, the ratio for the CO1 is much lower compared to 16S and 18S genes. So this clearly indicates like different genes from different genomes has a different ratio for DNA to RNA. And as the time passes, they clearly indicates the ratio is declining. So based on this ratio, we might be able to tell when the genetic material is released. Is it coming from a dead material, or it's actually coming from a live organism? Is it recent or past, based on these ratios? So here I would like to summarize a few points. The nuclear DNA and RNA have shorter half-lives compared to its mitochondrial counterpart, like as we saw, because of its structural differences. And we could actually predict eDNA release time based on the eDNA and eRNA ratios calculating. A multi-marker RNA approach clearly enhances our ability to differentiate, is it a interfacing of false positives, is it coming from a legacy DNA or it's actually recently released organisms. This will really enhance in understanding or estimating the abundances in many cases, not just for the Dreissena mussels, even including like the fish growth rate. And recent studies has focused on calculating the abundance of the fish and its life stages of the fish, all those things. Combining the multiple markers with the eDNA and eRNA combinations might really help to understand their life stages as well as their physiological conditions of the organism. Certain genes will express under stress and certain genes may not express under the stress. Based on the selection of the genes, we could definitely use for not just for the monitoring presence absence purpose, but in fact, the state of the organism, if they are undergoing any physiological stress or not, that might be really good to understand all those details. And also, like, we need to further calibrate. This is all based on the laboratory studies, like in the mesocosms, but this needs to be done like more in the field studies rapidly. So that's where we are in the coming summer. We're planning to do in CSMI, that is a cooperative science monitoring initiative. We are planning to do lake-wide surveys in comparison with the sonar graphs and divers, where they get the images of the mussels, because they are generally patchy in shape. You know, if you're getting a sample from near shore to offshore, how is this eDNA in eRNA signals will match? So ground truthing with actual conventional surveys will enhance our capabilities to use, like, apply these models, like in the for better predictions. So in the coming summer, we are working with colleagues at NOAA Global to do this, expand this study, which could be easily applicable and enhance the temporal distribution of these models. With that, I'd like to thank everybody for listening to the presentation. Thank you so much. I'd be happy to take any questions you have.
All right. Thank you so much, Rao. I was just contemplating your future research and thinking how we'll need to have you back for another presentation once you move into the next phase. So thank you so much for that interesting presentation. I encourage everybody here to begin typing questions into the chat if they have any. There are some questions that have already come in, which we'll go through. But before we get started, Rao does have a couple of poll questions for this audience. So Tracy, could you launch those? Awesome, there they are. Okay, so yeah, feel free to, while you listen in, go ahead and answer these poll questions, kind of help Rao out with his research. All right, with that, we're going to go ahead and jump into the questions. Let me scroll up here to the top. All right, so we had a question that came in from Carol. rRNA typically has a lot of secondary structure. Does that affect this?
Catherine, can you please repeat the question, if you don't mind?
Yeah, of course. Sorry about that. Let me say it a little bit louder here too. Okay. So this question is coming from Carol. rRNA typically has a lot of secondary structure. Does that affect this? This, I think, meaning your research. And Rao, if you need to see any of the questions, they are here in the general chat. I'm just reading them out loud.
Hi, Carol. I know, like, Nate was working with you before. So yes, they have different structures. Again, we have seen different kinds of like RNAs, like ribosomal RNAs, tRNA, mRNA. So based on their structure, their degradation will be faster or slower. For example, like the messenger RNA is much faster in degradation rates compared to ribosomal RNA, where it has like a different structure. So that's where we need to choose markers from not just one marker. Multiple markers will help us to use, like, you know, one could have longer half-life, one could have a shorter half-life. Based on their ratios, we could actually tell if it is really a recently organism was in that location, or it's like a legacy one, or it's actually like a day or few days ago. So that's the beauty of selecting multiple markers here.
All right, thank you so much, Rao. Moving down the list of questions, we got a lot of questions coming in, so folks feel free to continue putting them in the chat. We had Paul link the publication for this work, so thank you so much, Paul. Actually, when you all close out of today's presentation, you will automatically be redirected to that paper. Okay, so moving down the list, we have a question from Catherine. How will this work at the low concentrations of eDNA typically observed in the field?
So thanks, Catherine, bringing that question. One of the challenges is if we are looking for an invasive species which is rarely occurring in the environment, it's hard to find its DNA itself. In that scenario, we might have challenges with the RNA. However, you have seen that RNA concentrations are way higher than the DNA concentrations if the sample is like a fresh sample, or like if the organism is within that environment. So sometimes it might help. Again, if we are just monitoring presence of an invasive species where they are really low in concentrations, we may not be able to get enough signal because the organism may not be in exact location. We might be still getting for DNA signal. In that scenario, we may not be getting for RNA signal. But if this is not just for looking for the rare organisms, then the RNA will be really a key player here.
Great, thank you so much, Rao. Moving on to the next question from Anne. In a natural system, not mesocosm, animals would not be removed and would continue to produce nucleic acid products. How would this impact your ratios and estimates of when the material was produced?
That's a great question. So I know, like in the laboratory, we want to see how the degradation patterns are there. So based on like how the number of live and dead material or the number of organisms are there, we could still build on the ratios. That's where we are actually extending our experiments to see how the natural system would reflect the number of samples, even not just presence absence or the number they're continuously releasing, but also like other environmental factors might be like UV or other degradation patterns like from nearshore to offshore. That's where we're trying to do more intensive work with the phonographs and scuba drivers to get actual abundance and how this ratio is varying from place to place when actually the animals are there. We could still build on the ratios, but we still need to work more on to develop those things. I think maybe next year by this time we might have an answer.
Thank you.
Yeah.
More of a reason why we need to bring you back for another seminar, Rao. So I'll make sure to put that on my very long-term to-do list. It looks like Tracy put your email in the chat, so we do still have 15 more minutes for questions or comments, and there are still more that we need to go through here in the chat. So moving down, we have a question from Marilyn. A general question, do you think that eDNA degradation would be similar in marine systems?
Thanks, Marilyn, asking, like you're asking question is like, is eDNA degradation would be similar in marine systems versus like the freshwater systems? The chances are highly likely the degradation patterns would be similar. Of course, they are different materials and they have different factors, environmental factors, again, like UV or bacteria or whatever. But when it comes to the ratio, what we are looking for eDNA in the RNA is still we could build on this. again, these ratios has to be built for different environments and also for different species. So now we are building more for the Dreissena mussels, but if you're more looking into fish species, we have to do these calibrations again. So, but once the calibrations are done, that's the beauty is like we combine with the environmental parameters and we could make much better predictions. So to answer your question, there might be slight variations in the degradation patterns, but once we know what factors are, like what are the environmental factors, and then we could easily come up with the solution for that.
All right, thanks, Frau. Our next question is from David, and it looks like this question potentially was answered later on in the chat, so I'll just read the question from David and then the follow-up comment for it. So David asks, Rao, are you familiar with the very similar work being done by the Environmental Quality Operations in Austin, Texas? And then later on we had Gregory say, David, yes, we are familiar with EQO. Sigler is supporting them to test their approach in the Great Lakes. So Rao, I don't know if you wanted to comment on that. If not, I can move on to the next question.
Yeah, thanks, David, and thanks for answering for that. I think, yes, we are doing, Sigler is supporting, Sigler is the Cooperative Institute for Great Lakes Research. Yep.
Okay. All right, so our next question is from Kim. Thanks for an excellent presentation, Rao. I might have missed it, but can you use the various marker ratios to differentiate between a recent signal from a dead animal compared to the signal from a living animal that was deposited some time ago?
Yeah, that's the whole concept here, Catherine. Like, what we're trying to see is like, when we have different ratios, because as you saw, for the 16S, the ratios are different, and the CO1 ratios are different, and 18S, the ratios are different. When we combined all these ratios, we could say, hey, when is this material released? And if the ratio is like 3, we know that the material is released fresh. If the ratios are between like 2 or 1, we know this material is released like few days ago. But again, when we're looking at the messenger, if it is already, the signal is gone, we could easily tell that this is not like within a day or so. So that's where multiple markers are helpful for us rather than an individual marker. So it's telling whether they have the, that's where, if it is a dead, like, we know the messenger, like the H2B gene or something, they won't be producing anything or they won't be expressing, although they have their DNA counterparts, but RNA is completely gone, or they're not expressing it also. I think those are really helpful to make a differentiation from the live versus dead, or if the organism is there and it's moved to further different place a few days ago.
All right, thanks, Rao. A few more questions to work our way through. Our next one is from Jen. Is there a significant difference in methods or costs for eRNA collection?
Yeah, that's one of the major challenge for eRNA is the cost is huge. Although that could be drastically reduced, but more research needs to be done in that area. Right now, we are using the commercial kits where we have really good success. Depends upon how the quality of the RNA is extracted, your results might change a lot. So in this case, the kits we use are definitely expensive for RNA research. So basically, we could use like a trizol, like gold standard, like a more phenol chloroform method. But again, we need to optimize and make sure that we are not losing any of these RNA during the extractions or conversions. Right now, I would say definitely the RNA research is expensive than the eDNA. But we are working on a robust method development. If we have like our own method development, that the cost would reduce drastically.
Thanks, Rao. Okay, moving along here. We have a question from Joseph. Your linear relationship was drawn through some points with some widespread. What is the resulting confidence in estimating the timeline of a sample? In other words, what is the practical range of estimates for a given sample?
That's a good one. Again, here we had so many replicates and so many mesocosms parallelly running multiple samples. So the range is we could come up with a 0.1 within 0.1 to 0.2 range. But when we consider, again, if you're going from immediate time point, that might be huge. But when you look from a day 1, a day 0 to day 2 or day 3, you could clearly tell the differentiation is huge and they're significantly different. I think it definitely helps to differentiate from a live versus dead material immediately, especially when you have that messenger RNA incorporated into that. But differentiating within a few hours, it might be a little bit challenging. We need more work or more calibrations needs to be done.
All right, thanks, Raul. The next one is it looks like a comment from Chris. I'll read it out loud, and then Raul, if you'd like to respond, you can go for it. So Chris says, the second survey question can be interpreted in more than one way. It can be possible to assess the physiological state of organisms by looking at eRNA levels from different functional pathways, e.g. stress response genes versus routinely expressed genes.
Yeah, thanks, Chris. I appreciate your explanation. In fact, I think we should go back to poll questions and see how they responded and then what that means. So application of eRNA research, select all that apply, organisms detection could be improved. That is true. 38% agreed with that, and physiological state of the organism could be monitored, 28% agreed. And life stages of the organism can be monitored. As Chris was telling, yes, it's absolutely true. The life stages could be monitored as well as the physiological state, if they're undergoing stress or not. We could pick up the stress-related genes from the environment and could be monitored to understand not just the individual organism, whole ecosystem, if it is stress-related genes are highly expressed in that environment. That will be more useful. Our director, Greg Dick, and we're planning to do something, some sort of stuff in the future to see if we could pick up the stress-related genes in different environments to monitor if the system is going under stress or not. And in fact, 57% agreed that all of the above are correct, and this eDNA research could be applied to that.
All right, thanks, Rao. We're coming down to the last couple questions here. So you got a lot of accolades in the chat. Scrolling down here. We have a question from Paul. Wouldn't you expect the perfect ratio of a mesocosm would not be expected in natural environments because of the potential for legacy and introduce eDNA from non-local sources?
The mesocosms would be always better because it's more controlled environmental studies, but it doesn't mean that we can't predict this in the environment. I think that's where we are trying to do a whole bunch of lake-wide surveys and comparing with the traditional or conventional methods to see if the abundance is changing. Will these ratios change if they're spawning? You might release more amount of the genetic material. So is that leading to a different ratio versus a normal time, or perhaps a different genes, or are there any variation within them? I think it's going to be a little bit different, but definitely we could answer with that using the multiple gene approach.
All right, thanks, Rao. All right, we have a comment here from Henry, so I'll give you a moment to comment back if you'd like to. It will be interesting to see degradation rates in areas of different levels of eutrophy and UV radiation.
Yeah, that's one interesting thing, Hank. We'd like to see is like, that's where the nearshore and offshore experiments will come into play is like, if UV is penetrating to the deeper levels or like in the compared to the shallow area, would that have a higher degradation rates? Or if it is like more eutrophic, like where we see higher amounts of harmful algal blooms, like, how does this is going to impact? My guess is like the shallow areas, the degradation would be much faster than the deeper areas where the UV penetration is less. But again, we have to test this. Uh, there are some studies done with this kind of studies with the UV impact, but they are not really detailed studies comparing with the conventional methods hasn't done. I think with our summer sampling in the Lake Huron would answer that quite detailed way, especially we are going to have like all conventional comparisons.
All right, thanks, Rao. We got the final question here from Jeff, and there's actually a follow-up question from Tracy that kind of goes along with Jeff's question. So Jeff says, great presentation, thanks. Are you aware if this technique has been applied to hydroids or corals? And then Tracy asks, in addition to Jeff's question, will you try this next on any saltwater species?
I'm not aware if it is applied to corals, but again, this eRNA, eDNA technique is really a recent one. Very few studies has applied this study. There was a review out by Matthew Yates in 2021. They cited this as the only study where they actually done like quite a bit of comparisons with multiple markers. But I know like most of the people are working out. I think the publications could come out if not they are already there. I haven't if I haven't seen on the corals yet. I might have missed. Yes, this could be definitely applied to marine systems, and we are planning to collaborate with other NOAA labs to apply this to marine systems. Hannah.
All right, thank you so much, Rao, for your presentation, for taking the time to answer all of these questions. Anne had a question, actually, which I think I can answer, and I also answered here in the chat. Yes, the presentation was recorded, and it will be up on the NOAA Omics website. We also have an email address, noaa.omics@noaa.gov. So Anne, feel free to just email me directly, and I could just provide you with a link once it goes up on our website. All right, so with that, we're going to wrap this up. Rao, thank you so much for the fantastic presentation, and thank you everybody for joining today. We really appreciate it. The NOAA Omics Seminar Series generally takes place on the third Wednesday of every month at 12 p.m. Eastern Time. We actually, in the new year, have a few spin-off presentations coming up, so I will email out that information and ensure it gets distributed widely. So our next seminar will take place on January 19th, 2022, and will feature 2 speakers from NOAA AOML. So thank you everybody for joining us, and have a great rest of your day.
Thank you so much everybody for listening and attending.